Bismillah Rahman Rahim. Assalamu, dear students. As you know, this video is definitely about O levels biology. 5090 is the code of the paper. And in my last video, as I told you, that I am going to complete the chapter of transport in plants. So, students, I hope you are enjoying your life, you are good in health, and definitely you are happy. And after watching my video, you will learn many things. And I need your support as well, means your feedback. So kindly inbox me your feedback as well. So in today's lesson, we will cover these key points. Definitely translocation. What is the meaning of translocation? How translocation occur? What is the meaning of source and sink? And definitely we identify the phloem, source and sink areas with the help of diagram. We also discuss a little bit about xylem again, and we discuss about its structure as well. So let's start with the lesson. And first of all, we need to discuss what is translocation. In this chapter, we discuss transport in plants, the transport of materials. Materials included water and minerals. These materials transported from roots to the aerial upper parts of the plant in the leaves through the xylem tissue. And that one is known as transportation of liquid, transportation of water, and ascent of sap as well. And now the transportation of photosynthetic products, which are also known as assimilatives. The transportation of photosynthetic products like glucose, sucrose, amino acids, these products after photosynthesis, when they made in the leaf, transported from leaf via phloem tissue to all other parts of the plant body. So the areas who prepare, who make the food content, who make the assimilatives like glucose, sucrose, and amino acid. Obviously, these are green areas like leaf and some green stem areas are known to be the source. So, students, the leaf area is known as source. Why it is called source? Because source is the region where production of food takes place by photosynthesis. So leaf are the source. And what about the sink areas? The region, the areas of the plant where this food stored or utilized. And what are the sink areas? Definitely, first of all, root. Roots are the sink areas. Fruits. Fruits are the sink areas. And rapid growing regions where very rapid growth takes place, where the metabolism is really high. Those areas are also known as a sink. So, root, fruit, and growing regions are the sink where this prepared food is utilized and stored. In roots, definitely, food is stored, glucose is stored in the form of starch. In fruits, as you know, fruits are really sweet in taste. So food is stored, sucrose is stored in the fruits. And growing regions like new leaves where they are emerging flowers, fruits, new branches. So these areas where the development of new leaves takes place, branches, fruits, flowers. So you can add flower as well because flower is also sink because it use this food for the definitely growth. But important thing is this here that we need to understand the little bit structure of the phloem as well. Phloem is a tissue. Phloem is made up of specialized cells which are called sieve tube cells. So here sieve tube cells. Why these are called sieve tubes? Because you see here at the end of each cell of sieve tube there are the pores. These pores are known to be a sieve plates. And another some adaptations of sieve tube cells that they don't have a nucleus, they don't have a vacuole, they have many mitochondria. And along with the sieve tube cells, some other cells are present which are known as the companion cells. And these companion cells are really important because food translocated from the leaf mesophyll cells, palisade cells enter first into the companion cells. And as you see here in the companion cells and in the sieve tube cells, as you see, there are small gaps. These are called sieve plates. Uh, these gaps are known to be a plasmodesmata. So these are plasmodesmata. So plasmodesmata. So through these plasmodesmata, sucrose diffused in the sieve tube cells and loaded in the sieve tube cells. What happened next? Water, because I already told you that these two pipes are always parallel to each other. They are, as you see that the ball point, in the ball point, the pipe which contain ink, it considered to be a xylem, and another plastic pipe which is all around that one ball point, it is known as a phloem. So phloem is all around the xylem. Okay, so xylem is everywhere where phloem is present. So from the xylem, water enter in the sieve tube cells and the water potential of the phloem cells become lower, become thin. And because of this reason, when water pressure become high, water potential increases and water pressure increases in phloem now because the entrance of water from the xylem, water move in both the direction, upward and downward in the sieve tube cells, and water move maximum toward the sink areas. Now students, come to the other slide where I will show you this mechanism more clearly and visibly. As you see that the translocation, the movement of food in the form of glucose and sucrose, especially on amino acids, move from the leaf area to the other sink areas via phloem tissue. Here you see that pressure flow hypothesis and the bulk flow hypothesis. There are two hypotheses here, and both are the same because in pressure flow hypothesis also tell us that when food from the source cell is loaded in companion cell, as you see, and from companion to it diffused into the phloem, then water added, water added from the xylem and make the water pressure high in the region of this phloem sieve tube cells. So because of the water addition, the solution become thin, dilute, and move fastly upward and downward position in the sieve tube cells where the concentration of water is less, maximum toward the sink areas like fruits and the root. As you see here, this is the sink area where whole solute, glucose, sucrose, amino acids, They are diffused into companion cell, then into the root area and stored in the form of starch or in the form of other nutrients. And what happened at the end of the last one sieve tube cell? All the solute is uploaded in the companion cells, surrounding tissues, and remaining is water. So water return back into the xylem and again move from high water potential to low to the upward direction and again available here for adding in the phloem again. So in this way, we recognize pressure flow hypothesis is the hypothesis in which we discuss that how the high osmotic pressure, water potential, gradient drive make the phloem sap to move from one sieve tube cell to another sieve tube cell and at the end to the sink areas. And bulk flow because maximum quantity is moved. So dear students, this is about translocation, and here, according to the syllabi, this is another our component or learning outcome from the syllabi that we need to differentiate between the cross section or the transverse section of the leaf. So here we need to know the actual detail of leaf, stem, and root internal structure. So now I'm going to magnify the diagram one by one so it become easier for you to see the detail of each and every that part of the plant. So dear student, first of all, I want to show you the detail of leaf. As you see in the leaf, there's a upper epidermis tissues. It is cuticle, and these elongated shape are the palisade tissues. Okay, and in the center vascular bundle, xylem and phloem. In between, spongy mesophyll tissues, lower epidermis, along with the stomata and the guard cells. So this is the anatomy of a leaf. Now come to the stem. In the stem, if we see that in the stem area, that having same epidermis. After epidermis, there are cortex tissues. Here, sclerenchyma. You can write here cortex tissue, and the vascular bundle in the stem. They are in the form of circle. This is vascular bundle, vascular bundle, vascular, xylem, phloem. So xylem and phloem tissues collectively called vascular bundle, and these vascular bundles are arranged in the form of circle here in the stem. So this is the difference between stem and the leaf. At the top, you will see that in the leaf, vascular bundles are arranged one in the center, but here in the stem, they are in the form of circle. Okay, let's see what happened in the root. So students, as you see here, in the root area, the outer epidermis is there, and some of the epidermal tissues are having these extensions as well, which are known as root hair cells. So in the epidermis of root, extended root hair cells are present. Then in the center, cortex is same, and the vascular bundle is one and large in the center, which is star shaped. Red one is the xylem and then phloem, but its shape is star shaped. This is the recognition. How you can easily recognize the difference, or you see that it is stem or leaf or the uh, root. So we can easily recognize these three type of tissue because in the past papers we have number of questions. We showed that you need to recognize these three tissues and label as well. So here in front of you, tissue system, leaf, stem, and root. Okay, the next one is in the syllabi, tell about the some detail or adaptations of the xylem tissue. Why the thick walls are present. As you see here in front of you, there's a diagram of this tissue, which is a xylem tissue made up of vessel cells, which are dead and hollow, having thick walls. Why they have lignified thick walls? Because these thick walls allow them to withstand the high pressure generated during water transport and provide structural support. So 2 marks. 1 mark allow to withstand the high pressure generated during water transport, and second mark provide structural support to the plant. No cell content because xylem tissues are the dead tissues and they are made up of vessel cells, which are dead cells. No cell contents, the absence of no cell contents to make this tube hollow to maximize the space available for water flow. This reduces the resistance to the water movement and allow for efficient transport of water. 2 marks. Why these cells are joined end to end with no cross walls like two straws? As a straw, you use the straw to drink the cold drinks. If you connect two straws, three, four, it become a very long pipe. So as in the straw, both the ends are open. Here as well, in each vessel cell, and walls are open. So this arrangement forms a continuous, uninterrupted tube, minimize the resistance to water flow and allow for rapid and efficient transport over long distances. 2 marks. So overall in the summary, that in the past paper questions, that why the thick walls and what are the function, what are the adaptation, why the thick walls of xylems are there, why no cell content, why there no cross walls or end walls. So this is the role and the function of these adaptations which I told you. So dear students, today we finish this chapter transport in plants, and in my next video I will upload which is definitely about transport in animals, mammals. So inshallah, in the next video, I will explain you that one. Kindly give me your suggestions, give me your ideas, how I can further enhance or make these video more interesting, and what are the things which you want to be added. Your feedback is really important. Thank you very much. Allah Hafiz.